Okay, thank you everyone. Welcome to the session of evaluating the quality of tissue engineered products. And I'm Jane Lebkowski. I'm the president of Regenerative Patch Technologies, and I will be the moderator today. We're all excited about the prospects for tissue engineered products, but know that there are challenges going forward. And the developments in the field really spur on excitement of how to meet these challenges and look at implementing tissue engineered products as therapeutics in the future. So what we're going to do today is have a general discussion about some of the issues, some of the challenges that are going forward with looking at tissue engineered products. I will let my fellow panelists here introduce themselves, tell you over 1 minute about what they are, what their different organizations are focusing on, and then we'll launch into the questions. We'll start with Eric.
Okay, great. Thanks, Jane. Thanks to ARM for allowing me to be up here and chat with my esteemed colleagues. I am working with Aspect Biosystems presently, and my responsibilities there are driving partnerships and business relations. What we're doing at Aspect is creating tissue-based therapeutics through bioprinting, and our main focus at the moment is in type 1 diabetes and liver. But certainly, you know, the tissue engineering space has been around for quite a while, so it's going to be very interesting to have this chat. I think it's a great point of discussion, and looking forward to it. Thank you.
Geoff.
Yes, great. Thanks, Jane. Kevin Healy, certainly excited to be here today. Thank you, Jane, for moderating this, and to all the organizers of the event. At Enzyvant, I serve currently as the lead for regulatory affairs, pharmacovigilance, and quality assurance. In Enzyvant, we're focused on rare diseases and regenerative medicines. I'm going to talk primarily today about our experiences with rethymic, which is an allogeneic processed thymus tissue. So it's a product derived from unrelated donors of thymus tissue. And we obtained FDA approval for rethymic just over a year ago, on the 8th of October, 2021. And can certainly talk today and share some of the experiences we had going through the late development BLA submission, review, and approval process, and particularly with regards to focus on evaluations of product quality.
Geoff.
Great, thank you very much. Really appreciate the opportunity to be in front of everybody today. I'm Geoff Hamilton, the CEO of Stemson Therapeutics, and we're a preclinical stage company based here in San Diego, focused on leveraging induced pluripotent stem cells to regenerate skin-based tissues. And right now we're focused on our first few programs of actually regenerating hair follicle tissues to treat folks with alopecia and a lot of forms of hair loss. And it's a really, really exciting time to be a part of specifically the tissue engineering field as we look at a number of therapies now making their way through clinical trials to restore lost function in things like the brain or the pancreas or the spinal cord or the eye and other parts, and seeing that intersection of not just cell-based therapies, but also bringing in the biomaterials to help restore sort of the structure needed to establish these tissues and prepare them for transplantation is a particular area that I'm interested in, looking forward to talking to you all about today.
Okay, so, you know, one of the things that, a question I always get, and one I know that's important for the entire field, is if you're looking at a tissue engineered product, how do you measure potency? How do you measure that this structure is active? And maybe we can start off with Kevin sharing some of your experience of measuring potency, and have you had interactions with any regulatory bodies about how to measure potency and what might be ways to tackle that?
Yeah, certainly, Jane. We had extensive experience and interactions with the FDA in measuring potency for rethymic. Just step back. In general, the regulations are broad. Refer people to the FDA guidance on potency assays for cell and gene therapy products. There's a lot of really good information in that guidance. And the the key is to have a qualitative measure of product quality and relevant biological activity. So this can be direct biological activity or a surrogate measure of activity. And for Rethymic, the product development historically utilized a quantitative measure, a qualitative measure. A qualitative measure of product quality, a histology-based assay. And through the development and BLA submission process, we worked on developing a semi-quantitative measure. So making that more quantitative, which requires some significant method development and method validation and significant interactions with the agency. And I can certainly talk about those in some more detail, but maybe give others an opportunity to discuss quantitation in general.
Eric, any thoughts?
Yeah, I think obviously the release of product is absolutely critical, and having that quality measure of potency is very, very important. Sometimes it's a challenge though with tissue engineered based products, right? The concept that we're working with is we're creating a tissue, but almost by definition is going to remodel to some degree or mature within the body. So it's very difficult to have the exact measure of potency, but to Kevin's point, the ability to have a measurement that could be a surrogate of how that tissue would work is absolutely critical. So for type 1 diabetes, for example, we're looking at glucose-stimulated insulin secretion, and we're also evaluating technologies related to oxygen consumption rates. These are measures that will indicate the degree of health of these particular constructs that we're creating and give you an idea, but ultimately the remodeling within the body is going to be a very important aspect of the final functioning of an implant.
Geoff?
Yeah, I'll offer a point of view based on our own experience in that, Eric just mentioned early stage indicators of secretion factors that are certainly very important, I think, in most, if not all, tissue-based products. But what's really interesting is some of these tissue-based products we're trying to create, it's also about structural variation and structural patterns, that it's not just about what is secreted, it's also about the three-dimensional arrangement of the tissue that is critical in its functionality. And that's certainly true in our case when we're trying to create either skin or hair follicles. And what we're learning here is the secretion factors are somewhat ephemeral over time and change with stages. And what we're really interested right now in is this morphological development. What does the actual structure look like on a process that frankly can take sometimes months, either in vitro or in vivo, to develop into the fully functional outcome that you need with a durable functional tissue that's been transplanted. And you need some early indicators of that. You can't always wait 6 months for an in vivo outcome. You need an earlier batch release, and a lot of it can be literally the behavior, the pattern, morphological pattern of how the cells are arranging themselves in vitro. And we use a lot of imaging-based technologies to measure that, and we're trying to sort of correlate that to ultimately to in vivo outcomes. So that's an exciting area for us.
Yeah, so that brings on other questions about, what new technologies are you very excited about in looking at measuring potency, measuring structure, measuring function of the various tissue engineered products that we're looking at? Again, maybe Geoff, you want to expand on that?
Sure, absolutely. So new technology development in this space is critical for our success, and thankfully there's a number of folks working on this. I would say that the two areas of heaviest focus for us on new technologies is AI-based imaging analysis, which I'll talk a little bit more about, and then genomics-based, or particularly gene expression-based analysis, and how that changes over time. And so both of those data points we're collecting and building algorithms around those to be more predictive. And we really love, I mentioned imaging, you'll hear this a few times from me, we love imaging just because it doesn't have to perturb, per se, the cells in vitro. It can be quick, it can be fast, and if you have a validated algorithm that can sort of quote unquote certify the potency. And we actually have seen a few instances from some others like Kapil Bharti's lab, who is regenerating retinal pigmentation epithelial cells from iPSC to treat macular degeneration using an image-based system that has been validated through the FDA and is a batch release criteria for potency there. And so we're really sort of hitching our wagon to that development and seeing more and more of these AI-based technologies and algorithms come out to complement what has been an ongoing effort on the gene expression side, which I imagine many people have some experience with. So I would just sort of focus to those two areas showing the most promise at this time.
Yeah, Kevin.
Yeah, absolutely. Couple points. One, I would point out that in addition to being used for batch release, these potency assays can be important for detecting changes in manufacturing and also to support comparability assessments for future manufacturing changes or maybe improvements to your manufacturing process. So they're important in the use not only for batch release. With regards to technology and automation, we actually went down the avenue also of automated imaging for histology. Ultimately came into challenges with validating that method. And I would point out that it's an area where we can focus on continuous improvement. We had one assay that was suitable for product development. It was qualitative in nature. We tried to move all the way to a fully automated quantitative assessment and ended up landing somewhere in the middle with a semi-quantitative assessment that was still being conducted by pathologists in a scoring system. And what we had to do was establish a very rigorous scoring system and go through that full method validation, having multiple pathologists, ensuring accuracy and precision, and having the full set of SOPs, processes, detailed procedures that would be appropriate for a BLA, which is different than your typical pathological lab that's used in a hospital setting. So the use of that technology for us has been a stepwise progression.
Eric?
Yeah, I think as Geoff and Kevin both touched on, I think visualization is really, really important when we're talking about three-dimensional structures, and we're looking at that very intently. So we have our engineering team that is using machine vision to essentially track at 100% inspection, if you will, of the tissue that we're producing and printing, so that we can evaluate and make sure that we set our parameters for the various architectures that are critical to our tissue, and use that in a feedback loop to our production system so that we can control these parameters very, very closely. And we think this is really critical because during our Interact meeting, the FDA was mentioning that having a very solid understanding of your tissue is going to be absolutely critical. So we can now, as I mentioned, do 100% inspection and have the data on file essentially as part of the batch record for any particular implant that we're printing. So machine vision, and then using that in a machine learning capacity to be able to control your manufacturing process and have the quality data to back it up is something that we think is very, very important.
Hey, Jane, one quick interjection. I think it's worth calling out a technology we sort of glossed over and Eric referred to as the bioprinting technology.
Yeah.
This is critical, and I think we would be remiss not to call that one out as a sort of a basic capability to arrange structurally the building blocks of the cells that we're starting with and the biomaterials that they're combined with. And so the ever-increasing and advancing ability to do this more precisely, to do it in a more complex arrangements, to do it in arrangements that drive important aspects such as directionality of growth of the tissue itself, which can be important in function. We would be, I think, remiss in calling that out as one of the key base technologies here specific to tissue engineering.
Yeah, I totally agree. I was that brings up actually another question, which is manufacturing and the scalability of manufacturing of these tissue-based products. You know, what do you see? How are we going to scale up to make sure that, you know, right now manufacturing is more artisanal for many products, not all, but for many products. How can we look at bringing in what technologies do we need to do? What technologies do we need to qualify and validate in order to make these economically feasible? Go for it, Geoff.
Should I start? Okay. So when I think about tissue engineering, it's two steps. It's how do you make your cells and your materials, basically the base components of a tissue, and then how do you bring them together and integrate them and arrange them. And so I think a lot of the focus of a meeting like this and the sort of furthest advancements in the cell and gene therapy space are how do you program cells, how do you make cells. I don't think there's anything unique about our needs in that particular step that's different from a lot of what the rest of the companies experience in terms of consistent reprogramming of cells, consistent delivery of factors, or whatever technology approaches you use to generate your cell building blocks. I think it's really how you integrate then those components that we need as building blocks for tissue engineering, where we need some focus and advancement. I think the ability to move out of a human labor-based, hands-on approach at the bench, which is sort of where we are in many cases today, to better and better, more automated and reproducible capabilities that are robotic in nature. Bioprinting is one strong aspect of this that can scale. And I think the basis of that technology is out there now and needs folks to just continue to scale that for us. And I think the integration of inline QC is going to be really, really important in enabling the manufacturing and scalability, at least from our perspective, to be able to test as you go through the process and being able to catch early if a product is getting off track and maybe on its way to becoming a bad batch. Catching that earlier with inline QC is another area I'd call out. I'll let the others expand.
Yeah, Eric, why don't you go? You're involved in bioprinting companies, so.
Yeah, so automation is going to be key for any of the technologies we're working with, whether it's autologous and it's a CAR-T-based technology. Having the ability to reproducibly manufacture that is key. If it's an allogeneic material or tissue, like what we're doing, again, the same type of control is going to be key. It's all about the reproducibility. The challenge becomes when you're taking multiple components and putting them together at one time, right? So having the various technologies from a cell scale perspective. So if we're going to be needing, hundreds of billions of cells, it's going to depend, of course, on the lot sizes. But to combine that with your biomaterial in a manner that the cells can maintain their viability and their ability to be metabolically active eventually, into almost a continuous type processing situation where we can print one implant after another, is going to take a lot of engineering. So we're very, very fortunate at Aspect to have a very strong engineering team. And so as we're developing our early phase products, we're thinking about how can we manufacture and scale. So these the communication elements for the various components that are coming together, along with the machine vision, machine learning for inline quality measurements, are all top of mind for us as we're driving to develop these technologies on manufacturing processes.
Great, Kevin.
Yeah, absolutely. I would mention that some of the themes are really important, that reproducibility and consistency. Rethymic is approved for an ultra-rare indication, congenital athymia, in which patients are born without a thymus tissue. And there are about 20 patients per year in the US born with that condition. So for us, the scale-up was less critical. I think for all of these, tissue engineered products are so broad and diverse that everybody needs to think about, what are the critical parameters for them and in their product development. So for Rethymic, the key for us was to have sufficient scale at our contract manufacturer and the contract testing facilities to ensure that we would have appropriate staffing. There is a product being generated, it's one lot for one patient, needs to be highly synchronized and provided on demand. So ensuring that we would always have those capabilities within our testing facilities was really most critical.
Great. So one of the concerns or one of the challenges that we have going forward is looking at the durability of these products that we're looking at. How do they change after implantation? How long can they survive? How long can they function? So, again, opening it up to the panel, what are the approaches that you're taking to assess durability of your products? And, again, how are we integrating a lot of the quality assessments along with that assessment of durability? Kevin, you want to talk about that?
Sure. Well, as a late-stage commercialized product, we're in a different situation than others on the panel. So we have the benefit of over 25 years of clinical data and long-term survival data. With Rethymic, there's approximately 75% survival of patients who have received the product, and greater than 90% survival of those who've survived beyond the first year. So we have those as our primary measure of efficacy during the clinical studies. We also utilize flow cytometry as a measure of thymic activity to be able to assess that, both the development of thymic activity in the recipients, but also to see that it's sustained over time. We did, to try to, during the clinical development, utilize biopsies to evaluate tissue activity and the durability of that over time. However, there were many challenges associated with that. So with Rethymic, the Rethymic slices are actually implanted into the quadriceps of the recipient, and then as the slices were biopsied over time to evaluate viability of the tissue as well as thymopoiesis. And the challenge, there were multiple challenges associated with that, including even ensuring that you were able to correctly biopsy the right location. So that was one direct measure that we utilized in clinical development, but that was not a required aspect of the clinical studies, or wasn't required for FDA approval, and it's not required in the commercial setting.
Eric.
Yeah, I mean, it's a very important and critical question for some of these long-term implants, depending, if they're rare diseases in the liver space, people will depend on these devices for the rest of their lives, in certain cases. For type 1 diabetes, of course, the same thing. So we want to make sure that we generate tissues that have that durability. So how can we guarantee that? I think to Kevin's point, we do a lot of assessment of our materials in the models that we're looking at from a functionality perspective, and understand what's going on from a remodeling perspective, again, to assess that durability over time. I think one of the major criteria, though, is functionality. So does the patient maintain a degree of glucose homeostasis, for example, for type 1 diabetes? It's going to be clear if it's not working. You'll be seeing glucose rising and lack of control. So from that perspective, the functionality of the device lends to the understanding of the durability, and of course the intent is to work that out through early development, and ultimately getting into the clinic with patients, once we establish the criteria for the maximal durability that we can achieve. But ultimately it will be tested in the patient.
Great. Geoff?
Yeah, my point of view on this is I genuinely don't think that there are in vitro models that I've seen in many different indications, and certainly in Stemson's specific application area that can reasonably predict for durability. I wish there were. I wish we had all the resources to build one out. I just think there's no other way around just a good, well thought out, well designed, long-term in vivo study. Just got to face the music on that one. I don't think you can get around that. And I think selecting the right animal model, for example, is really, really critical in being able to establish a tissue that can survive and function, whether you want to do a human xenograft into some sort of animal, or I've seen others, and we've thought about this ourselves, about a homologous approach where you actually take the animal's tissue and you perform exactly what you would perform on the human tissue, except you do it to the animal's tissue and then transplant it into the animal as a kind of parallel, if you will, that you wouldn't take that into the clinic, of course. You would take your human approach to the clinic, but could be quite informative on what the potential durability is. And I think any company sort of facing this just has to build a plan that sort of smartly resources the funds and time needed to do some of these longer term durability. I think it's a really big challenge for our space to answer that durability question.
Terrific. All right, we have a little over 3 minutes left. I think what we'll do is just give everybody another minute to, what are your final thoughts on, tissue engineering products, the challenges we have in terms of assessing their quality and the steps needed to go forward in the future. We'll start with Eric.
Yeah, sure. Happy to try and give a closeout here. I think the paradigm of creating tissues has actually been around for quite a long time. I was doing some recollection of when Eugene Bell started doing his tissue engineering with skin. That was over 40 years ago now, and tissue engineering was probably really the first type of cell therapy. We've seen a lot of changes over the years. And the advent of stem cells certainly is now allowing tissue engineering to have a resurgence in the ability to scale. So it's going to be critical for the various tissues you can have. The other technologies from a gene editing or engineering perspective, where hypoimmunity might be provided to a particular cell type, but the necessity to control that is still going to be prevalent. We'll need to, at least in certain indications, control the way that cells are maintained within the body. So this resurgence of scale with stem cell related technologies, this hypoimmune space, and the ability to combine various materials to produce these tissues is going to kind of build on this industry that has been around for quite a long time. So it's very exciting. I think something that's going to really impact the field as well is this kind of organotypic culture, organoids and other technologies where multiple co-cultures, if you will, of cells will be demonstrated and have been demonstrated to show increases in functionality from a metabolic function perspective. So I see a lot of application here in the future, a lot of potential in the future, and I'm just very happy to be part of it.
Great. Kevin.
Yeah, it's definitely a very exciting time in the field. I would just leave it at 3 points. The first is there's so much technology available and so many possibilities. You can go in a lot of different directions, but also you can use tried and true methods. You have those base methods and you can build on those. Second would be to work with the FDA and other health authorities. They have the benefit of seeing development occurring across the spectrum. It was mentioned some feedback from an Interact meeting. We got extensive feedback prior to the BLA submission and during that review and approval process. So leverage those interactions as much as you can. And third, think of it as a phase-appropriate and continuous improvement perspective. We had one potency assay that was utilized during product development, another for the BLA submission review process, and now we have a post-marketing commitment to develop a more quantitative assay, to assess changes in manufacturing and support those comparability assessments.
Geoff.
Sure, I'll wrap it up with maybe something a bit complementary and slightly different than Eric and Kevin's comment. And in tissue engineering, you know, the industry at large, many of you here, we're getting quite good at making cell types. And what we need in tissue engineering is then an extra step where you have to think about integrating those components together and bringing in many cases, a biomaterial as well. And what we find in our own work is that when you make a cell type, and then once you introduce it to its sibling cell type, we have to work with several cell types to create a tissue, it changes its behavior. And in fact, some of these cannot function until they're in the presence of its sibling cell types, if you will. So this is a really sort of new and emerging capability to study how that works, how to control it, how to reproduce it, how to use and develop biomaterials that can help support and drive that development is something I would just call out to the audience as sort of unique in the tissue engineering space, and some of us are sort of right at the forefront of trying to figure out how exactly to reproduce and control that integration perspective for our products.
Great. Well, we've come to the end of our session. I want to thank the panelists for their participation and thank the audience for attending. Thank you.